Dear students, welcome to online classes, day 11th. Today we are going to discuss about absorption of the digested food material. Before going to learn this topic, let us recapitulate the previous content. In last class, last lesson, we discussed about mechanism of digestion. In the topic, the digestion of carbohydrates, the digestion of proteins, digestion of lipids, fats, and digestion of nucleic acids. All these are macronutrients. All these macronutrients, in what way they undergoes digestion in our alimentary canal, as sequentially we learned in last class. Okay, right, students, we are going to learn about subsequent events of physiology of digestion. In the protocol of digestion, we completed all these things: ingestion, digestion in buccal cavity, swallowing process, peristalsis, and digestion in stomach, and digestion in small intestine. So in this lesson we have to cover absorption of digested material, assimilation of digested material, and defecation. So these three things to be covered in this lesson. All right, next, now we are going to discuss absorption of digested food material. What is absorption? Absorption is nothing but it is one of the process. For what purpose? In which what happens, the digested food material passes through mucosa lining of alimentary canal into blood or lymph. So whatever particles, whatever micromolecules, whatever liquefied material produced by the digestion, that material to be passes into the cells of the mucosa lining of gut. Afterwards, from that mucosa lining, it enter into the blood or lymph. That phenomena is called absorption. Various parts of alimentary canal mediates absorption phenomena. The pattern phenomena completely different. Some areas facilitate absorption of some variant of content, and some parts of gut facilitate the absorption of other substances. So the absorption phenomena is completely different in different regions of the alimentary canal. Let's see in what way absorption takes place. You observe this visual. All these are micromolecules. The micromolecules are moving to cells to then into blood or lymph. So in this fashion, the absorption to be mediated. Now, absorption by oral cavity. So first, the absorption starts oral cavity onward, buccal cavity onward, it will start. Of course, without digestion, the macronutrients never undergoes digestion, of course. But other than macronutrient, some micronutrients also we consume through the food. So such micronutrients to be absorbed in different regions of our gut. So you observe in oral cavity, whatever absorption occur, that is completely passive process. Generally two processes remain. One is active transport, another one is passive transport. What is active transport? Active transport is nothing but in which if energy utilized, energy consumed, energy spent, So that type of transport is called active transport. Suppose in this transport, if energy doesn't spend, energy doesn't consume, so that is called passive process, passive transport. Follow? So whatever absorption occurring in our buccal cavity, in our oral cavity, in this phenomena, no energy to be spent, no energy consumed. So it considered as passive process. Followed. So through oral cavity, what contents to be observed? You observe this Vishwa. Drugs. The drugs also absorbed into blood through the lining of the lower surface of the tongue region, lining of the tongue. Followed. Right next, salts, some electrolytes, some salts are absorbed into blood through oral cavity. So in oral cavity, drugs and salts to be absorbed. Followed? Right. Next, after oral cavity, then the subsequent part of our gut is stomach. Stomach also mediates absorption phenomena. So, what contents to be absorbed by stomach? You observe this visual in what pattern the absorption mediated by the stomach, larger size food particle going to break down into micro molecules, and after that some of the contents to be absorbed into the wall of stomach. So in here also the absorption is passive process, otherwise through osmotic gradients to be mediated from high concentration to low concentration. So in this process, what content to be absorbed? Same, drugs and water, salt and simple sugars, alcohol, all these five contents to be absorbed into blood across the wall of the stomach. Follow, students? So if we compare the oral cavity comparatively with oral cavity, you observe in stomach the surface area is more, rugae are there. Such rugae increases area of absorption surface. That's why comparatively with oral cavity, in stomach more number of contents to be absorbed as passive process manner. So what are the contents to be absorbed across the stomach? They are drugs, salts, simple sugars, and alcohol, water. Follow? Right. Next, the absorption in small intestine. If we observe small intestine, the small intestine is the major site for absorption. Approximately 90% of absorption mediated by the small intestine, only 10% by stomach, oral cavity, that regions. So 90% of absorption achieved by the small intestine. If we observe this small intestine, in this small intestine, the mucosa lining of the small intestine gave rise numerous foldings. Already we learned it thrown numerous foldings into lumen of the intestine. Such foldings are called intestinal villi. You observe this visual. Foldings are there. And you observe this villi. Visual foldings are there. They called villi. On villi, again the cells of villi shows microvilli, brush border surface. So that increases area of absorption surface by which the intestinal villi plays major role in absorption. Followed. Right. Next, in what way absorption mediated by small intestine? We'll observe. Beneath the villi, blood vessels, blood capillary network, where artery ends, from there vein starts. So blood capillary network executed. In the same way, lymphatic capillary network also executed. So the lymph vessels are called lacteals. All these things we learned in previous classes. So blood capillary network and as well as lymph vessels, lacteals also executed in the villi. So these things to be facilitate the absorption phenomena. Right, you observe this visual. In this visual, clearly blood vessels, blood capillary network is there, and lacteals also present. You observe these are capillary network, and this one you observe this is lymph vessel. These are cells of villi. So all these facilitate absorption phenomena. So what things to be observed? You observe sugars. The sugars are produced, monomers are produced by digestion of carbohydrates, monosaccharides, glucose, galactose, fructose, like that. Sugars and amino acids that produced by digestion of proteins. All these to be absorbed into the capillary network. They absorbed into blood vessels of the villi. Remember, very important, the sugars and amino acids to be absorbed into blood capillary of the villi. Next, the digestion of lipids results fatty acids and glycerol. So the short chain fatty acids to be absorbed into blood, but the long chain fatty acids unable to absorb into blood. So the long chain fatty acids and as well as glycerol molecule to be absorbed into lacteals, lymph vessels instead of blood vessels. Remember, ask in entrance examination, the fatty acids and glycerol to be absorbed into lacteals, the sugars and amino acids to be absorbed into blood capillaries. Followed. So with this we got idea about the content that absorbed in each part of the villi of the small intestine. Let's you observe this visual. This is villi in which capillary network lacteals are there. So sugars, amino acids, short chain fatty acids are absorbed into capillary network, blood vessels into the blood. In what pattern they absorb? The sugars are absorbed by facilitated transport. So with the help of sodium plus ions, with the help of carrier ions, by utilizing the energy, by facilitated transport, the sugars to be absorbed into blood vessel. In the same manner, the water to be absorbed on basis of osmotic gradient. Followed high content to low, high concentration to low concentration, like that. So next, amino acids by secondary active transports. In the secondary active transport, the amino acids are coupled with sodium plus ions. By that sodium plus ions, calcium plus ions, through carrier ions, all these amino acid sugars will be entering to the blood vessel. So that is called facilitated active transport and secondary active transport. Okay, so the facilitated active transport and secondary active transport results the absorption of the sugars and amino acid. Next, long chain fatty acids. The long chain fatty acids are unable to absorb into blood vessels. They absorb into lacteals, lymph vessels. In what way? It is a lengthy process in which various steps to be involved. The first step, formation of mycellae. Second one, endocytosis of mycellae and formation of chylomicrons and exocytosis of chylomicrons, and next event, absorption of chylomicrons into lacteals and separation. You observe this visual. Ultimately, the chylomicrons to be enter into the lactea. So it is a lengthy process in which 5 steps to be involved. The first step is formation of mycellae. In what way mycellae to be produced? Let's discuss. Observe. The mycellae formed in the lumen of small intestine, that is the location. In what way? The long fatty acid chains aggregated together, assembled together with bile salts, and phospholipids also participated. So the phospholipid molecules, long fatty acid chain molecules, and bile salt molecules aggregated together and form a mycellae. You observe this visual. In this visual, long fatty acid chain molecules are there. These long fatty acid chain molecule going to aggregate with bile salts. You observe these are bile salts, glycolates and tarcholates, and next they combined with each other, joined with each other along with phospholipids, and ultimately they produce a separate compounds. These are mycellae. You observe this is mycellae. These mycellae are hydrophilic compounds. Before that they are hydrophobic. Now these are hydrophilic. Followed. So now these mycellae, after formation mycellae, these mycellae undergoes endocytosis phenomena. Whatever mucosa lining cells are there, with the help of microvilli, they intake the mycellae into the protoplasm. So you observe this visual. These are mycellae. These mycellae across the plasma membrane, they dropped into cytoplasm of the cell. So that phenomena is called endocytosis. After formation mycellae, they completely enter into the cytoplasm of the cell. Right. Next, third phase is formation of chylomicrons. After entering into the protoplasm in the mycellae, after entering the mycellae into protoplasm, now the endoplasmic reticulum processes the mycellae. It releases the some contents, glycoprotein molecules. Such glycoprotein molecules are aggregated, assembled around the mycellae molecule, and gradually forms a new molecule, that is chylomicrons. So chylomicrons to be produced in the protoplasm of the mucosa cells. So the mycellae turn into chylomicrons. Followed? Right. You observe this visual. After entering into the mycellae, the endoplasmic reticulum going to release the protein molecules. Such protein molecule aggregated, bind with mycellae, and formed a new molecule, that is chylomicron. You observe this is chylomicron. Followed, students? Right. So after formation chylomicron, what happens? After formation chylomicrons, you observe this is a separate visual of chylomicron. Glycoprotein coat is developed. The envelope is complete glycoprotein coat. So there is chylomicron. Next, after formation chylomicrons, exocytosis takes place. What is exocytosis? From the cell, the formed chylomicrons move out. That is exocytosis. After exocytosis, it leads to absorption of chylomicrons into lymph vessels. Lymph vessels nothing but lacteals. Followed. So you observe this visual. In this visual, the chylomicrons going to absorb into lymph vessels. So that is absorption of chylomicrons into lacteals. The green color vessel, lymph vessel, lacteal. Right. Next, you observe this visual. One more visual. In this visual, the lymph vessels, green color vessel, lymph vessel is there. The chylomicrons absorbed into the lymph vessel. Right. Next, with this absorption of chylomicrons into lacteals completed. So after entering chylomicrons into lacteals, through the lymphatic system, the chylomicrons are circulated in lymph. So in our body, in the vascular system, the lymphatic system is accessory route to the circulatory system. So in lymphatic system, lymph to be circulated. In our body fluids are, one is blood, another one is lymph. Two kinds of body fluids are there. Blood circulated, blood flow in blood vascular system, lymph circulated in lymphatic system. In this visual, the lymphatic system organization is there. So the lymph circulated in lymphatic system. Whatever chylomicrons are absorbed into lymph vessel, they to be circulated in lymphatic system. After circulation, finally these chylomicrons are released into our venous blood. In what way they released into venous blood? You observe this visual. So chylomicrons, green color vessel you observe, yellow color droplets you observe. Chylomicrons enter into lymph vessel. Through the lymph vessel, they are circulating in lymphatic system, and ultimately they empties into venous blood. Followed. In what way they discharged into venous blood? The chylomicrons relieved into bloodstream through thoracic duct to left subclavian vein. Thoracic duct is major lymphatic duct. So first they relieved into thoracic duct. From the thoracic duct they relieved into, they dropped into left subclavian vein. Followed. So after relieved into left subclavian vein, now the chylomicrons circulated into blood. In the blood, in the venous blood, lipoprotein lipase, you remember, very very important, lipoprotein lipase enzyme is present. This lipoprotein lipase enzyme acts on chylomicrons and break down the glycoprotein envelope that present around the chylomicron. So in previous slide we observed the chylomicron structure. Around the chylomicron a glycoprotein envelope is present. So that glycoprotein envelope breakdown lysis lysed by lipoprotein lipase enzyme. So the lipoprotein lipase enzyme disintegrate that glycoprotein envelope, by which whatever long fatty acid chain molecules are present in that chylomicron, that long fatty acid chain molecule, phospholipids and other things to be relieved into the blood. And now through the blood they move into the tissues. So first they move into the liver. In the liver they will be participate in lipid metabolism. Whatever comes under lipid metabolism may be utilized in the synthesis of lipids, and the excess amount of lipids will be stored in adipose tissue. In case of starvation, they will be utilized in oxidation process, beta oxidation process, and produce what? Energy. So that is the phenomena of the lipid absorption. So It is a lengthy process comparatively with sugar amino acid absorption. The lipid absorption required more changes. Followed. So the lipid absorption include formation of mycellae and endocytosis of mycellae and formation of chylomicrons, exocytosis, entering into lacteals and circulation and relieved into blood and breakdown of glycoprotein and ultimately the fatty acid released into bloodstream. Followed. So with this we completed absorption of lipids and now absorption of absorption by large intestine. Through the large intestine you observe drugs, minerals, water and bacterial synthesized vitamins to be absorbed across the large intestine. Followed. So with this we completed absorption phenomena. After completion absorption, the subsequent event is assimilation. What is mean assimilation? Assimilation include various changes. The digested food particle reaches the tissue through the blood. It is one of the event. And next, such particles become integral components of protoplasm. In the protoplasm, they become integral components and they used for production of energy and they involve in catabolism process and produce energy and these contents also used for growth and repair. So all these changes, all these events togetherly considered as assimilation. Follow? So after completion absorption leads into assimilation in which what happens? The digested liquefied food material reaches to tissue, become integral components in protoplasm and used for production of energy and used for growth and repair, that is assimilation. Right, next the final event is deification. Undigested food material, unabsorbed macromolecules passes into large intestine. So such undigested food material, such molecules are held together by the mucus that's secreted by the large intestine and the entire material is called roughage, otherwise fecal material. So this roughage and fecal material are retained in large intestine. Already we discussed through the large intestine some amount of drugs, minerals, whatever vitamins present, water that to be absorbed across the large intestine and the remaining roughage and fecal material, the temporarily it's stored in rectum and afterwards it expelled out by anus. That phenomena is called egestion. Followed. So the defecation is nothing but expulsion of the roughage or fecal material through anus is called defecation. So you observe this visual. In what way? The roughage to be moved along the large intestine and finally expel out through anus, clearly explained, that is egestion. So it requires powerful peristaltic movements that are mediated by the muscle layer present in the rectal canal and various reflexes involved in the egestion process. So with this we completed total events that